A-Level Biology 9700 Paper 4 Variant 2 May June 2024 Question number 7 8 and question number 7 is a student investigated the effect of temperature on the rate of photosynthesis at two different light intensities. The rate of photosynthesis was determined by measuring the rate of oxygen production of the plant. And here you can see a graph in which the rate of oxygen production is shown on the y-axis, whereas the temperature is on x-axis. Two curves has been shown. One is at low intensity, low light intensity, other is at high light intensity. So question with reference to figure 7.1, describe and explain the results of the investigation for the plant in low light intensity. In low light intensity, you can say uh, the a constant uh, rate of production of oxygen has been shown. As the temperature is increasing, there is no change in the rate of oxygen production. So we are going to write the answer in the light of this graph. That is, the rate of oxygen produced or the rate of photosynthesis is low or it stays constant. The production of oxygen produced is low or constant. Constant as the temperature is increases. So we can say that if light intensity increases, the rate of oxygen production can also increase. So we can write that in this case, the light intensity acts as a limiting factor in this case. In this investigation, you can say that. And next question is, with reference to figure 7.1, describe and explain the results of the investigation for the plant in high light intensity above 30 degrees centigrade. We will move towards the graph again. You can see the rate of oxygen production is optimum at 30 degrees C, and after 30 degrees C, it is decreasing. So it is decreasing, and at 50, it is stopped. So we will write the answer in this way, that is, the rate of oxygen produced is maximum at 30 degrees centigrade. So after 30 degrees centigrade, it start decreasing. Oxygen production is maximum at 30 degrees centigrade, and after it start decreasing. So we can say the reason can be because the temperature is optimum, then the reason can be the denaturation of enzyme. due to denaturation of enzyme, the reaction or the production of oxygen stopped, decreased or stopped. Next question. Describe the process that produces oxygen during non-cyclic photophosphorylation. And students, you know what happens in non-cyclic photophosphorylation. And you know the non-cyclic photophosphorylation is a process which results in the movement of the electrons in a non-cyclic way to synthesize the ATP molecule by using the energy from the excited electrons that are provided by photosystem 2. So we are going to write in this way. So when light energy is absorbed by the photosystem 2, is absorbed, photoactivation of chloroquine takes place, photoactivation takes place, due to which photolysis takes place. And what is photolysis? Breakdown of water molecules. By the water splitting enzyme. Water is water breaks into protons, electrons, and oxygen. Question number 8. Question number 8 is figure 8.1 is a diagram of sensory neuron. Name the type of the cell represented by cell A. So you can see the movement wave impulse will be in this direction. So due to in this direction, we will be these are the A are the receptor cells. So they are the receptors. And next question is that forms a synapse with the structure B and synapse. You can say this is the sensory neuron and the nerve impulse will move from sensory neuron towards the relay or intermediate neuron. And name the cells that form the myelin sheath. Schwann cells. So state and explain the differences in the transmission of impulses between myelinated and unmyelinated neurons. So in myelinated and unmyelinated neurons, there are difference. You can say the impulses the movement of the will be faster or of nerve impulse will be faster, and you can say the we will explain in this way that the myelin sheath insulates or prevents movement of ions. The function of myelin sheath is to insulate the axon, but all areas of axon are not covered. So the points where there is no myelin sheath in the axon, those points are called nodes of ro- nodes of Ranvier. Are the points where there is no myelin sheath. So these are the gaps where there is no myelin sheath. So what happens over here? Action potential, when action potential will jump from node to node. From node to node. So in this way, the action potential will takes place from sensory to intermediate, and from intermediate to motor to effectors. So in this way, the nerve impulse will move, and the differences between the unmyelinated and myelinated sheath will take place. Now the next question is, opioids are the therapeutic drugs that are used to relieve pain. Morphine is an example of an opioid. It affects the functioning of the synapse. Is a diagram of the chronergic synapses, and what happens in them. That is the diagram. You can see the pain stimulates from one axon, and you can see presynaptic neuron towards the postsynaptic neuron. So you can see the opioid receptor cells are present and on the presynaptic neuron, and on the postsynaptic neurons there are the receptors which are used to open up the uh, channels for ion exchange. So I would like to we will move A, B, and C. So these are the three different points. With reference to figure 8, describe the difference between ion channel A and the ion channel B. And you can see the ion channel A are present on the presynaptic neuron, whereas the other B is present on the postsynaptic membrane. So we will write the answer in this way. The A is on presynaptic neuron and is a voltage gate gated, and B is on post synaptic neuron, which is non voltage gate. So B opens in response to binding of acetylcholine. A opens in response to uh, in response to action potential. Other words, we can say they opens when the action potential is reached over there. And next question is, morphine binds to the opioid receptors in the presynaptic membrane. This stops the ion channel A from opening. And J for opening, he suggests and describes the effects of the binding of morphine to this opioid receptor on the functioning of the cholinergic synapse. So what happens? So what happens in this case? That is when there is A open in response to action potential, when there will be when it will be closed, when it will not be open. So what will happen? There will be no action potential on that case. So what will happen as a result? That is the no calcium ions will not enter into presynaptic neuron. So vesicles containing acetylcholine will not fuse with presynaptic membrane, and when they will not fuse with the presynaptic membrane, so no neurotransmitters will release into synaptic cleft. And they will not bind, and the neurotransmitter will not bind with post-synaptic membrane. So channel B will not open for the ion exchange. No action potential will occur. Next question is morphine also binds to the opioid receptors in the uh, postsynaptic membrane. This results in the opening of ion channel C, allowing potassium ions to diffuse out of the cell. Shows how does it affect. So allowing potassium ions to diffuse out of the cell. So when there will be the potassium ions have so what happens when this morphine potassium ions will diffuse out of the cell? So there will be they have the positive charge. When they will go out, so there will be more negative charge inside, due to which the membrane will hyperpolarized, and due to hyperpolarized the it will not meet with the threshold. So no depolarization of the membrane will take place. So we are going to write these points. The potassium ions will move on opening of channel C, ion channel C. The potassium ions will diffuse out of the cell, causing less negative inside. So the potential difference will increase so threshold so in this way it will affect the synapse. Now it was all about the question number 7 and 8. Please like, share and subscribe my channel. Thanks for watching.